So this is a lecture on genomes, and in this lecture we're going to be discussing what a genome is, as well as what we kind of know about the human genome from studying it since we sequenced it in 2003. We're going to talk a little bit about the number of genes in the human genome and the different types of DNA that we find throughout our genome, as well as some kind of correlations or lack of correlations between how big genomes are, how complex organisms are, and how many genes that they have. And so first off, I want to start by defining what a gene is. And so we all know that the central dogma of biology is that DNA contains information that is transcribed into an intermediate called RNA, and then ultimately translated into proteins that we can see. And so we used to define genes as a sequence of DNA that encodes a protein. But now that we know more about our genome, we know that genes can also encode not just proteins, but functional pieces of RNA, like the tRNAs that are used in translation. And so we've sort of redefined the term gene in more recent years to mean a sequence of DNA that is transcribed into a functional RNA. Now, whether that means a functional RNA, like that stays RNA, as the tRNA does, or a functional RNA that ultimately becomes a protein, this definition encompasses both things. And so the complete set of all of these basically functional units of DNA in an organism is known as its genome. We tend to put that suffix OME onto a lot of different words to signify that's like the complete set of something. So a genome is the complete set of genes that an organism has. You can also study things like the complete set of RNAs that are present in an organism. And that would be referred to as a transcriptome because RNAs are transcribed, hence transcriptome. We tend to study transcriptomes not necessarily at the level of a whole organism, but in a specific cell or tissue type. We can see which genes are on, which genes are off by looking at the RNAs that are present in that cell or tissue. And we can actually manipulate conditions around that cell or tissue and see how the transcriptome or that set of expressed genes or RNAs changes under different conditions. And we can also study the complete set of proteins that's encoded by a genome. And that's referred to as a proteome. So genome is at the DNA level, the full set of genes. Transcriptome is the complete set of RNAs that are made in any given cell or tissue from that DNA. And then proteome is the complete set of proteins that are made from that genetic information. And we usually study proteomes at that tissue or cellular level as well, rather than over the whole organism. And so when we start to look into genomes, or all of the DNA contained in an organism, we see certain types of DNA sequences, or A's, T's, C's, and G's, right? Nitrogen bases, that are either repetitive or non-repetitive. So non-repetitive DNA is DNA that doesn't repeat, right? So self-explanatory. These DNA sequences are unique, and they only appear one single time in the genome. And so say that sequence is ATA, adenine, thymine, adenine. In non-repetitive sequence, you would only find ATA one single time in the whole entire set of DNA in an organism, or the whole genome. And there's also some sections of our genome that are made up of repetitive DNA, or DNA sequences that appear over and over and over within the genome. And so what we know from studying our genome is that the sequences inside genes that code for proteins, or genes that will ultimately become a protein, are non-repetitive. So each protein coding gene has a unique DNA sequence that only occurs one time within that genome. And that makes sense, right? Because why would you have repeated genes within the same genome? You have a unique sequence, it encodes one protein, you don't have to repeat that sequence again. And what you can see over here is actually different amounts of repetitive versus non-repetitive DNA in different organisms. And so you can see that in E. coli, a simple organism, they have entirely non-repetitive DNA. Every part of an E. coli's genome is unique, only appears once. There's no repetitive DNA at all. And as we get more complicated in organisms, what we notice is that now all of a sudden that non-repetitive DNA percentage goes down as the organisms increase in complexity. And we see more and more of that repetitive DNA in all these organisms here. And so there's like two different types of repetitive DNA. There's some that's considered moderately repetitive, where you might see it 10 to 1,000 times within a single genome. And then highly repetitive would be a DNA sequence that's repeated thousands and thousands of times. And if we actually look over here again and break down the moderately versus highly repetitive numbers, what we'll see is that there's actually an increase in both of them as organisms increase in complexity. And so things that are often found in the moderately repetitive DNA sequences are usually things that have secondary structures, like a tRNA would be encoded in a repetitive portion of DNA. Highly repetitive sequences tend to be regulatory sequences. And so the thing that's interesting about the increased non-repetitive DNA in a more or in a less complex organism as compared to a more complex one is that it seems like we're actually reusing a lot of these same genes, and then we're adding additional regulation as we increase in complexity, rather than increasing the number of genes per se that actually make a protein. And so how do we figure out like where a protein coding gene is in a genome? First, we need the entire sequence of the genome, right? We can take that sequence, all the A's, T's, C's, and G's, and we can look for specific trends or like sequences that will help us identify where a protein coding gene is. And so what we look for when we're looking at this whole sequence of the genome are the presence of exons and introns. And so every protein coding gene in its DNA is going to contain these two things, exons and introns. Exons are the sequences that ultimately end up in an mRNA and become translated into protein. Introns are sequences that exist, but are spliced or removed out before that mRNA is made into a protein. And so we're able to see here in this image, this gene has 3 exons and 2 introns, exon 1, 2, and 3. And then in the mRNA that's produced, what we see is only this exon 1, 2, and 3. Introns have been removed or spliced out. And that means that those introns will never be made into protein, but all the exons will. And so when we're looking for protein coding genes, we're looking for what's called open reading frames, which is a DNA sequence that has a start sequence or a start codon at the beginning, and then no stop codons all the way through until the very end. Right? And so here we're able to identify basically protein coding genes by looking for these stretches of sequence within the genome. There's a start codon at the very beginning, a start sequence that we know a protein coding gene has to have. There's a long open reading frame or a long stretch of DNA with no stop codons, meaning it's probably going to be made into a protein. And then there's one stop codon, singular, at the end that would signify the end of that gene. And so we don't do this by hand or by eye. We use computer programs to do this. They're able to scan through the entire sequence of a genome, and they're able to find these DNA sequences that are flanked on one side by a start one side by a stop and have no stops in between. And they're like, we predicted there's a gene right here because we see there's a start. We see a long sequence of DNA with no stop codons. It's not repetitive. And then we see a stop codon at the end. We're going to identify this as a gene that will make a protein. Like I said, highly dependent on computers and computer algorithms and bioinformatics. And so what we have gleaned from understanding protein coding genes, how they're structured, and from looking at genomes of different organisms or eukaryotes, is that we know the genome size and the number of protein coding genes is going to vary pretty widely between different organisms. And so this graph here shows genome size on the x-axis and the number of genes on the y-axis. And so you can see S. cerevisiae, which is yeast down here, has a relatively small genome size and a relatively small number of genes. If we increase in complexity and we look at C. elegans, which is a nematode worm, it has a larger genome in size and it also has more genes. But then what we notice is if we look at something like Drosophila melanogaster, which is fruit flies, definitely more complex than C. elegans, we see that they do have a larger size genome, right? Because this is moved down the x-axis, but they actually have less genes than C. elegans. And then here, Arabidopsis, which is a plant, even more complicated, they have a smaller genome than the fruit fly and a huge number of genes. Mice and humans, you can see over here, have a relatively large genome as well as a large number of genes. But what we notice is that mice actually have more genes than humans, even though they have a smaller size genome. And rice is just this complete crazy outlier. Rice has a relatively large genome and a huge number of genes, but if we think about it in terms of complexity, rice is definitely less complex than humans. So it's interesting that their genome is has more genes than humans do. And so ultimately, this leads us to this statement that the size of the genome in how many base pairs there are, how many A's, T's, C's, and G's, does not always correlate with how many genes that an organism has. There's not always going to be a relationship where a bigger genome means more genes, right? Because in the case of humans right here, they have a bigger genome than rice, but they have fewer genes. So that relationship is not always going to be correlated. And in humans, we know that our genome size is about 3.2 billion bases or 3.2 billion nucleotides, encompassing our full genome. And we have about the current estimate is 20,000 protein coding genes that will actually go from being DNA to functional proteins.